Hi class, in this recording we're going to start talking about genes and proteins, or at least begin that discussion. So first, let's talk about genes. When we think of a gene, genes are, here we go. Genes are packets of information, and this information is going to be stored in DNA, and this information is going to control for the production of a molecule of RNA. And we use that RNA to synthesize proteins. When we look at a protein, proteins are polymers of amino acids, and the order, the arrangement of the amino acids within the protein is determined by the nucleotide sequence in the DNA. So ultimately, the arrangements of A's, T's, C's, and G's within the gene determines the arrangements of amino acids within that protein. So here is an excellent figure that helps us show the relationship between these things. We have DNA being wrapped around histones to make a nucleosome, and then they wrap around themselves and wrap around themselves and wrap around themselves until we can see a chromosome. Now when we look at human beings, we have 23 pairs, 23 chromosomes per one set, and we have two sets of chromosomes. This genome within the human being is referring to nuclear chromosomes. We're ignoring mitochondrial chromosomes or mitochondrial DNA when we make this definition. As we look at the overall length of the genome is 3.1 billion nucleotide pairs long. Human beings have 46 total chromosomes in most cells, two sets of 23. Because there's two sets of chromosomes, we refer to that as a diploid. We as humans are diploid organisms. The abbreviation for diploid is 2 lowercase n. One set of chromosomes came from mom. The other set of chromosomes came from dad, one from each parent. Now as we look at our body, we make millions upon millions of proteins from just 20 different amino acids, and coded for by just 4 nucleotides. Using just 4 little pieces of information and 20 different building blocks, we code for millions upon millions of different structures. The way we do that is using a method, a language known as the genetic code, where we take those 4 nucleotides and code for the amino acids. When we think of 20 amino acids, the minimum number of nucleotides we need to represent or symbolize all 20 amino acids is 3 nucleotides per amino acid. If it was a 1-to-1 ratio, since there's only 4 nucleotides, we would only be able to represent 4 amino acids. If we had 2 nucleotides for the amino acids, 4 times 4 would allow us to represent 16 amino acids. So we have to bump it up to the next higher level. 4 times 4 times 4 gives us enough variation to represent all 20 amino acids. When we look at the DNA, these 3 nucleotides within the DNA is referred to as a base triplet. It's the 3 nucleotides that code for 1 amino acid. And then once that code is transcribed over to messenger RNA, we refer to those 3 nucleotides as a codon. A codon is going to complementary base pair with the base triplet in the DNA. So here's the genetic code. Please, oh please, do not memorize this. If you have any test questions, when you have a test question on this, I will give you the codon cheat sheet. Notably though, I want you to focus on this MET. MET is the start codon. This start codon is the very first codon for every protein. It tells us when to begin transcription and translation. So as we look at this, methionine, MET, is the very first amino acid in every single protein. Also notable, if we have the AUAG or UGA, those are the 3 stop codons. Please memorize that AUG is methionine. How do I know AUG is methionine? We have first position, second position, third position. So first, we have A. So everything in this row is an A. And then for our second position, I'm going to go with blue for second position. Everything in this column is U with the second nucleotide. So everything in this box is A and U for the first two nucleotides. And then for the third position, let's go third position in green. We have UCAG. Methionines at the bottom of the 4. So, and we have the G down here, which I covered up in red. Let me make a G for you again. There's our G in third position. So A, U, G corresponds to that methionine right there. And that's how we read the genetic code. So if you were to be given an amino or a codon, let's say you had a U for your codon, and you had to figure out what the amino acid is. You start with U for the first, U for the second, and then U for the third, and know that PHE, and you probably all you would say is PHE because you haven't memorized this yet, but phenylalanine is the UUU codon, or I should say the UUU codon codes for phenylalanine. So during this process, we are going to synthesize proteins, and we need to synthesize proteins in all body cells, except for a few of our sex cells and a few of our immune cells, which all will contain identical genes. Different genes are going to be activated in different cells at different times throughout the cell cycle, and also in response to different stimuli. Any given cell that we are going to be looking at will use about 1/3 to no more than 2/3 of its genes. The remaining genes are going to be remaining dormant, and that's okay because by activating or deactivating different genes, we can differentiate ourselves. We can take a muscle cell and make it a skin cell or make it an intestinal cell. So it's good that we don't have all genes active all the time. When we're synthesizing proteins, we activate the gene. An activated gene is used to make messenger RNA, also known as mRNA. That messenger RNA is complementary to the gene, which means that it will base pair to the gene. And then after that messenger RNA is produced, it's going to migrate or travel from the nucleus through the nuclear pores to the cytoplasm. Within the cytoplasm, it's going to code for the amino acids of the protein. During the process of protein synthesis, we start with DNA. That DNA is used to make messenger RNA, and that messenger RNA is used to make a protein. This is known as the central dogma of biochemistry. And this process of protein synthesis can be broken up into 2 subcategories. For instance, the first category is transcription. The root word here is script or scribe. And during transcription, we copy the information down. We don't change the language per se. We just make another copy. And we go from a nucleic acid to a nucleic acid, from DNA to messenger RNA. Transcription is going to happen within the nucleus of the cell. And then during translation is the second step. And this is when we go from one chemical language to another chemical language. You translate, kind of like going from English to French or from Spanish to Russian. And in this case, we go from a nucleic acid, messenger RNA, to an amino acid, which is going to be our protein. And translation is almost always going to occur within the cytoplasm. That's all we have for this recording. If you have any questions or comments, please feel free to post them on the class discussion board or shoot me an email. And as always, happy studies.